We continue to talk about exam 3, lecture 2, the microbial genetics. This is a template. We talk about genetics, which is a central dogma. So what we have here is the DNA goes to RNA. That is called transcription. And DNA itself will happen is replication, and the RNA will become a protein, and this is called translation. And the whole thing, we call this a central dogma. It is a blue plan when we talk about genetics. And we have already talked about the replication and transcription. So today we want to try to, replication and transcription, today we try to get to the translation. And this is what you can see. We mentioned already. The DNA is a double strand. One running opposite direction, 5' to 3'. Another one 3' to 5'. And they will synthesize the mRNA. And why we say it is one of them a coding strand, another one is template strand, because they are based on the gene complementary policy. A complement with T, G complement with C. So you have this RNA. However, if you look carefully, this mRNA sequencing and the coding strand is exactly the same, except you don't have thymine, you have a uracil. Okay, that's why this is called a coding strand. Now, when we move on to the translation, we need a special RNA, which is called a transfer RNA. Okay, so we want to get to these things later, the structure of tRNA. So we want to talk about translation. So translation, we are using 3-letter codon to talk about that. Or we just say 3-letter code. So how many gene base we have? A, U, G, C. And if it's 3-letter, we have a box 1, box 2, and box 3. This is box number 1, box number 2, and box number 3. So we have a first math question. If we have A, U, C, G, 4 different type of gene base, and we have 3 boxes, theoretically, how many possibility will form if it's a 3 letters codon? That's a very simple math, a high school math. How many possibility in the box 1? Could be A, U, G, C, anything, so it's 4. How many possibility in the box number 2? It is also 4. How many possibility in the number 3? It is also 4. So we multiply them, we equal 64. So theoretically, there is a 64 genetic code. However, we know how many we have. It's 61. Why? Because there is a 3 stop codon. Okay, does not mean anything for translation amino acids. That's UAA, UGA, and the UAG. And this table tells you about that. It stopped the codon right on the top. And every time when we do the translation, the first one is always AUG. It's methionine. Is that right? Translation to methionine. So this is some basic knowledge. Now we come out with that. We talk about this central dogma. Ideally, there should be exactly the same thing happened for translation, transcription, translation. However, more or less, you're going to have something, mistakes happen or errors happen, will cause mutation. So therefore, you have a box number 1, box number 2, and box number 3. The rule has been set very flexible. We were very strict for the first box. We also very strict for the code in the second box. But we give you flexible for the code in the third box. This comes out a very important phenomenon called a wobble. Which means, if my first one gave an example is A, and the second one is G, then the last one, that could be anything, could be A, U, C, G. They maybe represent the same amino acids. Because of this wobble impacts, we have 20 amino acids. And we have a 61 genetic codon. What happened is redundancy will happen, which means one amino acid could be translated by more than one genetic codon, could be 1 or 2 or 3. And that is, you look very carefully. For example, leucine. Did you see that? CUU, CUC, CUA, CUG, all translation is leucine. Which means CU in the first and second box is very strict, but the last box could be A, could be U, could be C, could be G. So that gives you flexible. That's called a wobble. This gives you some space to do the mutation. And those type of the mutation will not impact the final translation of the amino acids, will not impact or later on become a peptide or protein. This is called a silent mutation. So it's very magic. Okay, that's something we want to talk about, the translation, before we go. Okay. Now, by the way, do you know the structure of amino acids? You learned in your other classes. Center chiral C, we have NH2, we call it an amino group. We have a carboxy group right here. And we always have is H. And on the other side, we have all side chain. Now we talk about 20 amino acids. The difference is the all side chain difference. So we give you some of the example. We're not going to list all 20. That's going to take 10 minutes. So for example, if R is H, that's the simplest amino acid is glycine. But be careful, if it's glycine, then it's not a chiral C. Is that right? Because H are the same. If R is CH3, that's alanine. If R is this guy, what is that one called? Phenylalanine. And if the R side chain is CH3, CH2, CH3, CH2. If the R side chain is CH3, S, CH2, CH2, this is methionine. We got more complicated. If the R side chain is CH2, 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 and go C, NH2, NH3 plus, what we call this guy? Lysine. And this is called epsilon amino acids. With this long chain, what will happen? It's very easy to happen, the reaction with glucose or maltose or reducing sugar will happen, the Maillard reaction. So that's why the milk powder, you store it at room temperature for extended period of time, they will become yellowish. So these are things we can talk in chemistry class, we just skip it, okay? So these are the amino acids. What I do is represented by the 3 codon system, and they give you a flexible, which is a wobbling, which is we call the silent mutation. Now, once we talk about this translation, we cannot avoid to talk about the important and the very special chemical is tRNA. What this means? That's a transfer RNA. So what is the structure of the transfer RNA? That's very special. Like that. We call it a clove leaf shape. And we have a 5 prime, we have a 3 prime. Then this is most important. Those are the 3 letters. Yes, corresponding to the mRNA, we call this is a anticodon arm. Now you have these like D arm, variable arm. Sorry, D arm is on the other side. The D arm is right here. You have a D arm, T theta C arm, and also variable arm. So what happened is, if I give you a whole genome sequencing, how do you know it is tRNA? How do you know it is transfer RNA? If we break down every single piece of the gene base, you will see there are some very special, gene base. It's not only ATGC. You see the PY, PU, theta, T comes out. If you see some very weird or very special base comes out, like theta C, like T, a TM, these comes out, that's possibly is a transfer RNA. Okay, so we need to get to know the transfer RNA first. This is very interesting. We always talk about that. Amino acid activation. So what this means, what's the amino acid transfer means? This sounds very confusing, the picture here, and because it's upside down. So you don't understand this one very well, but I can put on the other way, you may can understand it more clearly. So don't forget about, regardless of how complicated, we say the gene, the nucleic acids, regardless of RNA or tRNA, it's a very boring chemical because the major part is 5-carbon pentose. It is a 5-carbon ribose. And on the other side, you have a base. Could be A, G, C, U, so we call this side is a base. And this is H, this is H, this is OH, and you go here, you have H, OH, you go here, you have H, CH2, so you go here, you have a phosphate there. So where the phosphate's going to connect it? The phosphate later on going to connect it with this guy, with the tRNA. So if you draw this way, will makes your life much more easier to understand. And that's 5 prime, and that's 3 prime. Is that right? That's tRNA. Now, where's the amino acid gonna go? Goes right there. How they connected? So we're gonna redraw it again. How they connected? You have H. Is that right? You have NH2. So sorry, NH2 right here. You have R1. You have H. NH2. Where are they connected? They lose water. Once they lose water, what they become? That's easy. That's what it is, looks like. And then you connect it here, you add another OH, add a C, add NH2, that will be R2, this will be H, these reacted, lose another molecule of the water, so becomes what? Becomes O, C, O, C, R1, H, become NH, CO, then go C, R2, H, NH2. What is this one called? Peptide bond. Obviously, this is double. So that's what it is. If you wanted to know the structure, that's more important. That's more clearly what happens. But what happened in the slides, in most of the genetics class slides, why it is so confusing? Because it is upside down. If you turn around, you will be more clear about that. The connection of the phosphate connected to the anticodon of the tRNA, and then you see the 3 carbon. The 3 carbon, the 3' carbon, that's connected to the amino acids. And of course, this need an amino acid. They need a tRNA. They need an ATP, the hydrolyzed ATP, and need a magnesium. And this process needs a specific enzyme, which is amino acyl tRNA synthetase. And we know the enzyme is very specific. So we have 20 amino acids. How many of these syntheses we need? At least 20. Okay, so that's how we understand these slides. Let's give you some of the example of the transfer RNA. So often in the exam, we ask people a very simple question. What's a tRNA carrier? tRNA is carrier, not a codon. It is an anticodon. mRNA is carrying the codon. Okay? Now, the next thing is very simple, is a very basic question. Where the translation will happen? Which machinery, cell machinery, will happen translation? Then everybody should tell me very clearly, loudly and simple, it's ribosome. So what's a ribosome looks like? The ribosome briefly looks like that. And the ribosome is a place to happen that is RNA translation. And they have 2 subunits for bacteria. The big one, we call it 50 S subunits. The bottom one is 30 S subunits, but when you add up them together, it's 70 S. It's not 80 S. Why? What S means, the Van der Berg unit. This is to measuring the velocity and the speed of the precipitation. It's not it's a chemical or physical index units. It's not just a simple mass. So that's why you have a 50s, 30s. You combine them together, become a 70s instead of 80s. Now, the very important thing we can be recognized here is not only that, the big or small subunits inside, there are some other things. So remember, we talk about RNA. What do we talk about? We talk about mRNA. That is for codon. We talk about tRNA. That is carrying anticodon. And we also talk about RNA. What does R means? That's ribosome RNA. So ribosome RNA, for 50S, there's one called 23S RNA. And in the 30S, there's one called 16S RNA. Those are important for the translation. Because how complicated it is, it doesn't matter. Once the mRNA come in, what's the key thing for this mRNA? Does not mean every mRNA will start to do translation. You need something can be recognized by the 16S RNA in the 30 subunits. The very special gene sequence is AGG. We call SD sequence. Full name is called Shine Dagano sequence. Once you have AGG, the mRNA will start to do the replication. So, start to do the translation. No. We have a slides, we can talk about that. We're gonna not draw everything. Look at here, 16S RNA is a ribosome binding site. It's binding to Shine-Dagano site on mRNA for protein synthesis initiate, and will become an amino acid tRNA. And we also mentioned inside of the 50S subunits, there's a 23S RNA. This is for peptidization, which means you have an amino acid coming. We need to become a peptide, which is a long chain or short chain of the amino acids. And you always remember, regardless what type of the amino acid sequence you start to do the translation, the first one is always methionine. But keep in mind, that is for plants, animals, and human beings, which is eukaryotes. How about the bacteria? Bacteria are a little bit special. It's called N-formyl methionine, which means a little bit of transfer here is that. N-formyl methionine. Archaea, eukarya using methionine. Okay, that is always the first RNA. Now this is basically is a template you can see. Start from the top. This is we talk about is transcription. We mentioned already, and this goes from translation. Okay, Shine-Dagano sequence when you know what they started to do. Now how they do? We don't want to be these complicated. Okay, I'm not going to draw it again. So what happened is, at the beginning, there is a tRNA. We'll carry on something and coming. let's say AUG comes. Once the AUG comes, they basically need energy. The energy is GTP. So GTP will hydrolyze with GDP and phosphate. But when you do the initiate, same as lots of the biochemistry reaction, they need an initial factor, 1, 2, and 3. And their function are different. So when you can see here is, originally, the initial factor 3 is attached to here. Okay, right here is the initial factor 3. So make sure at that moment, the 30S and the 50S subunits are separate. But once you started to do the translation, what happened? This will gone. This is going to take it out. Take it out by which one? Is initial factor number 1. Then the initial factor number 1 will attach to the side of the 30th subunits. And then this guy gonna come in. When this guy come in, they need to pay money. That's why they hydrolyze GTP, become GDP and phosphate. But how they come? So you need somebody to take them in. Is that right? This is very similar like you go to some island. You need some of the sheep, or you need some of the ship to go to a boat. So the boat up here is initial factor 2. So then they come. Okay, once they come in, it is interesting. There are 3 locations. And inside here, there are 3 locations. What are the name of those 3 locations? That's called the EPA. That's not the Environmental Protection Agency. We look at the news every day, they talk about that. That stands for different. What is the E means? Exit site. What is the P means? Peptidal site. What is A means? Amino acid binding site. So once they come in, where they go? They go the first right here. Then they start to move. And where the amino acid go? It goes here. So they start to move. The move is also a synthesize. It is always from 5' to 3'. We can see here how that goes. Okay, we jump in here. In my major class, this is I usually draw the whole blackboard, spending another 20 minutes to write the whole thing. But we're going to be a little bit brief here. Can you see the translation start? This tRNA carry on the MET, which is methionine comes here. Where they do start coding AUG, and then the second one coming. Okay, once the second one coming, what they do is this going to be insert here. And then this one will move to the E site and then release. So once they move to this direction, go to the E site, it's an exit site. Then the tRNA will be released. Then they're going to find another amino acid to attach to it and it goes back. So you can see the chain will be long and long. And here they need an enzyme, which is amino acid synthesase, to let the peptides become longer and long. And here they also need another enzyme, which is called transpeptidase. So synthesize, transpeptide, both happens. And then the chain become long and long, long and long. Okay? So that's what happens. Then finally, how do we know it will be stopped? It's relatively simple. It's not like transcription. They need a role factor, they need a stem, this stem loop, recognition of signal. They do not need here. We just mentioned about the stop codon. If they are finally find UAA, UGA, and UAG, either one of that, it will stop translation. And then what happened? The whole sequencing will released. And that release need a releasing factor 1, 2, and 3. Same thing like here, the last one. They need a releasing factor 1, 2, and 3. So you can go back to see here is that in these slides, it tells you very clearly what's the function of the EPA. E is the exit side. Empty tRNA, it leaves. A is the amino acid binding site. It's binding incoming amino acid tRNA. Then what the P means is peptidal site. It binds initial tRNA or tRNA attached to the growing peptides. Those are the three positions. And then later on, how do we know it's going to stop? Because we met the stop codon. It could be UAA, UGA, or UAG, and then we'll try to release it. Because those amino acids will become a peptide, become a protein, they have their function. So they need to release it, and then what's the release they need? Is release factor 1, 2, 3. And the whole process, we need a hydrolyzed GTP as an energy currency. And then later on what happened is the 50S and 30S subunits originally was combined during the translation. After translation finished, they will separate. Then everything will goes back. What happened is initial factor 3 will go back to attach to the side and make sure they are separated. Because we just mentioned when they started the translation, this will be taken out by initial factor 1. The initial factor 1 goes here. And then you have an mRNA coming, the mRNA have AGG short Agartho sequence, then they started to do the translation. And the translation, they need the initial factor 2 to carry on. Then they go to EPA, those 3 positions. Okay, so that's a very briefly introduction, talk to you what we mean about the protein synthesis, which is a translation. So we very briefly talk about here, spending about 35 minutes as part of the last section of the genetics part. Okay? So next one, the next big section, we want to talk about the biochemistry. Okay. Some of you now coming from the major of the biochemistry or biology, so we want to make sure this section is also as simple as possible, then you guys could be understand well what's going on here. So we talk about genetics, and then we're going to talk about the biochemistry. And biochemistry is a big topic. We're not going to go over too much detail. So we have this opening slides, which is mentioned, the chemoheterotrophs. So from the metabolism standpoint, there are lots of the terminology to describe the bacteria using or microorganism using energy. So for example, you have a bacteria using light as an energy resource. We call it a light heterotrophs. And some of them using carbon dioxide as an energy resource. We call it autotrophs. But most of the bacteria, what we talk about so far, E. coli, Salmonella, Listeria, Streptococcus, Staphylococcus, they are all we call chemoheterotrophs. It's a terminology for that. And during their life cycle, they basically is going through aerobic respiration, anaerobic respiration, and fermentation. Now, you can have these slides. These two slides you can combine using a very simple way to understand, which we talk about electron transfer. Okay, so what this means? Electron will be transferred, which means energy transfer. You can understand about that. Now, when the electron transfer, they always is from donor to acceptor. So regardless of how complicated those two slides we talk, we can simply to introduce to you a very simple way to understand. If the electron donor is an organic chemical, and the electron acceptor is also organic chemical. This process called fermentation. Electron donor is organic chemical. Electron acceptor is also organic chemical. That's called fermentation. If, this is the first scenario. Second scenario, if the electron donor is organic chemical, and the electron acceptor is inorganic chemical, this process called respiration. Then you look at here, they said aerobic respiration, anaerobic respiration. What this means? If the final electron acceptor is an inorganic chemical, it is oxygen. This is called aerobic respiration. If the final electron acceptor is an inorganic chemical but other than oxygen, for example, phosphate, this is called anaerobic respiration. That's a very simple way to understand about these things from the electron standpoint. Okay? This is what this slide's for. It looks like it's complicated. What is aerobic respiration? Final electron acceptor is oxygen. But you need to know oxygen is an inorganic chemical. What it means anaerobic respiration? It is a final electron acceptor is also an inorganic chemical, but it is not oxygen, it's others. But regardless of what, during aerobic respiration, anaerobic respiration, even fermentation, what we need to generate ATP, which is an energy currency, because otherwise a bacteria or microorganism cannot survive. That's why we want to talk about these things. And we want to talk about first is fermentation. Then we may get to the others next week. Okay, so fermentation. What is fermentation? You feel about fermentation every day. You like beer, you like wine, You like yogurts, those are fermentation. So what means fermentation? We talk about the electron transfer, we talk about the terminology already. Electron donor, organic chemical. Electron acceptor, also organic chemical. Okay, what is the example? There are basically two different types of the fermentation. The first one is pyruvate go to lactic acid. This is called hormone fermentation. But there is a second one. If a pyruvate become aldehyde, then aldehyde become alcohol. This is called heterofermentation. Do you know if the alcohol continue to do the fermentation, what it will become? Will become vinegar. If they continue to do. We will talk in the next 2 weeks. When we get to the last section of the lecture, we'll talk about the manufacturing of the vinegar. It's a very interesting topic. So this is the first thing you need to know. We skip the chemicals. That's pyruvate, that's lactic acids. Pyruvate, aldehyde, and ethanol. Okay? So basically you can understand that the first one, what happens? Yogurt looks like that. What's the second one? When the alcohol comes out? Red wine could be the example. Okay, so these are the two very simple examples talk about the fermentation. Couple of questions later. How many energy created through fermentation? How many ATPs? Very simple. Only 2 ATPs. How they created? Through substrate level phosphorylation. And this 2 ATP generate. It is coupled with glycolysis. So the things we talk about here, we need to mention what we're going to say here. We have a glucose. We're going through reaction, which is a branch reaction. Generate a product called a pyruvate. Then the pyruvate, we're going through as a transition step to become a acyl-CoA. Then the acyl-CoA goes here is citric acid cycle. Will generate a huge amount of the ATP. We'll talk about the calculation. But the pyruvate could go this way, which is going through fermentation. Therefore, it comes out with hormone and hetero. But what's the idea of here? To generate ATP. It's a very simple, very easy idea. This tells you what we talk about. Look at here, the pyruvate goes to lactic acid. Then it goes here. It's very interesting, the formula. We want to talk. Pyruvate become lactic acid. It's important. But how many ATP generated? We just say only 2 ATPs. Do you know how many ATP they created through this guy? 38, 36. Is that right? You learned in your class. Huge. That's only 2 ATPs. But how bacteria can survive? Did you ever think about that? Only 2 ATPs generated, but why bacteria can survive? Because at the same time, this guy comes out. The NAD comes out. What's that? Nicotinic adenine dinucleotides. It is an electron carrier. And when this comes out, what happened? They could accept a new electron here, or new proton here. Then they go ahead to do anything. Here is a very good example, which the electron carrier should be recycled during bacteria lifetime. You always remember, that is what we get to the bottom. So we talk about here, this is glycolysis. If you only do glycolysis, do you think bacteria can survive? No. Because there's no recycled NAD. So if bacteria to survive, they must do this. Or this. Because this will create the recycled NAD. That's a key thing here. And you can see here, that's what happens. NAD could be transferring. Can you see it? Glycerol 3-phosphates, that's one component.